Hey there, welcome back for the third and final installment of this set of notes. We're going to tackle the nucleic acids. Get this set up. Okay, so nucleic acids are made of CHOMP. Okay, so carbon, hydrogen, oxygen, nitrogen, and phosphorus. Hopefully you remember from your regular bio days that the nucleic acids are made of nucleotides. Those are the monomers, right? Nucleic acids or nucleotides are the monomers, and then a nucleic acid is made up of a sugar, a phosphate, and a base, right? Those three things. Well, if it's made up of a phosphate, then you have the CHOMP part up there. So you shouldn't get it confused with the protein side, even though it was CHONS. Hopefully you remember that the phosphate plays a really, really important role in nucleic acids. So it always has to go over here. So definitely the molecule of life, right? DNA and RNA are, they hold all the instructions, right? This is what governs all of our abilities of forming the body and functioning and all of the living things, whether it's us or a plant or a mushroom or whatever it is. So as I mentioned earlier, the monomer is a nucleotide, and a nucleotide is made of a 5-carbon sugar, a phosphate group, and a nitrogen base, which we abbreviate as ACG and T. The chemical names are adenine, cytosine, guanine, and thymine. Okay, so there's our pretty double helix over there. So DNA are huge, right? Really big molecules, and they're found in every single cell in your body. If we took the DNA out of your cell, out of any one of your cells, you'd put about 6 feet of DNA on a table. Okay, so 6 feet of DNA wrapped up into every single cell in your body. That's pretty crazy to think about. Okay, so they're very long, and they have this cool double helix structure. And here's what DNA actually stands for, if you don't know, which is deoxyribonucleic acid. And then the purpose of DNA is twofold: store the information that we all have to run ourselves, and then pass it on. Okay, heredity. So DNA is a polymer composed of nucleotides, and it's made of the 5-carbon sugar, the phosphate, and the organic base. So this is what one nucleotide would look like. Okay, so you see the phosphate group, see the sugar there, and then the organic base is the part that's interchangeable. That could be the adenine, cytosine, guanine, or thymine. That's all interchangeable over there. Okay, so grab the pen over here. So if we look at this, you can see phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. These are all nucleotides, right? So I might, if it would let me draw a line, maybe it'll let me draw one. Nope. So picture, here's one nucleotide, if you can follow my cursor. Here's the next one. So there's 4 nucleotides stacked on top of each other, and they each have the different base. Like you can see, the adenine is the double ring, guanine's a double ring, cytosine and thymine are only single rings. We'll talk about those in a second. A particular bond name, though, is phosphodiester. Okay, this guy right here, phosphodiester. So it's this line right there. Okay, so that links one nucleotide to the next. Okay, that line links the sugar of one nucleotide to the phosphate of the other. Okay, so a particular bond name in nucleic acids that join the nucleotides. Just like peptide bonds join amino acids, we have a phosphodiester that joins the nucleotides. Okay. So, mentioned this earlier, there's 4 organic bases. 4 organic bases: adenine, guanine, cytosine, and thymine. Okay. Uracil, you may have heard of. That's an extra base. That's a 5th base. It only occurs in RNA. Okay, RNA has uracil and does not have thymine. You probably remember swapping T's and U's in regular bio in some of your activities. So we're going to look at these single ring and double ring structures. So pyrimidines are organic bases that are single ringed. Okay, so here are your pyrimidines: cytosine, thymine, and uracil. I have no idea why I remembered this, but somewhere, someone along the way told me you cut pyramids. Okay, the cut being cytosine, uracil, and thiamine, and the pyramids being for pyrimidines. So I don't know why that actually is a saying, but it does help me remember. So maybe it'll help you, maybe not. But you cut pyramids. Hopefully that helps you remember which ones are which. I'll give you another way here in a second too after we talk about purines. Okay, so purines are just adenine and guanine. There's only two. Okay, and one of the ways I really remember these things, the difference between purines and pyrimidines, at least by looking at them, is purine is the shorter word, but it's the bigger structure. So purine, short word, double ring structure. And if I go back one, pyrimidines, longer word, smaller structure, just single rings. Okay, so as far as the length of the word and the size of the molecule, they're opposites. Okay, so maybe that'll help you remember what's what a little bit. Okay, so if we build a DNA, you build it this way, right? Nucleotide. And so we have what's called orientation here. We have the 5' end up here and the 3' end way down here. Okay, and the reason for these is if we look at where these are, so carbon 1, 2, 3, 4, and there's actually a carbon here that you don't see. So this phosphate is hanging off the number 5 position carbon, if you remember how we did those numbers. If you look down here, carbon 1, carbon 2, carbon 3, here is, and it doesn't show you here, and I wish it would let me draw it, but it won't. This is an OH down here hanging off the bottom. It's a hydroxide that dangles. So there's a hydroxide that dangles off the 3' end of the DNA, and there's a phosphate that dangles off the 5' end. That's how you can always tell which end of the molecule you're looking at. Okay? So we use those for orientation. Okay? So here's how, if you don't remember how complementary base pairing works, remember T and A always bond together. They form a word, at. And so they form together like that. Notice it's one single ring structure and one double ring structure. That'll become important too. And they have 2 hydrogen bonds between them. Okay, so those are complementary base pairs. A and T are complementary base pairs, and thus C and G are complementary base pairs. Again, one purine and one pyrimidine forming that pair. Okay, but in this case, 3 hydrogen bonds, not just 2. There's 3. Okay. So if we were to look at this, if we had one side of the DNA, we should be able to match it up, right? Because now we know what goes with what. And so we bring them in the other side here. Okay, we can see exactly what goes with what. And notice that on the other side, now we have the 3 and 5 over here, but they're opposite of the 3 and 5 over here, right? Because the DNA strand runs like this. Okay, so these are parallel, but they're running in opposite directions. Do you guys remember the word for that? That would be anti-parallel. Okay, anti-parallel, they run parallel, but they're running in different directions. So 5 to 3 on one side and 3 to 5 on the other. Notice the sugar is even upside down. It's pointing down on the right-hand side. Okay, so every living organism, no matter what it is, has these ACGs and Ts, and they're what have all the instructions, and they dictate the amino acids, right? This is called the central dogma of biology, and you guys definitely would have studied this in regular pre-AP, whatever you took. So DNA makes RNA, makes protein, and that makes all of these different things what they are, okay? Much more on this later in the year. Many more details. Complementary base pairs, as you would expect, except, there's a mutation there, right? So if we have a mutation, then that might lead to the wrong protein, and then you have a problem, kind of like what I showed you in the protein section. Okay, so real quick about some of the scientists that made this happen. On the far left, you have Watson and Crick. They were building the model. And then you on the right-hand side, you have Maurice Wilkins and Rosalind Franklin. And hopefully you remember this picture as well from your earlier bio class, this picture down here. That is called Photo 51. And it's a very famous photo that was taken by Rosalind Franklin. Okay. She was the only woman in the group. All of these scientists worked in the exact same building, and it was kind of the race to figure out the DNA structure. And Rosalind took this photo with a technique called X-ray crystallography. And Watson, who's the young kid over here, this guy over here, I'm putting the green dots on, that's Watson. He's the only American in the group. And he was only 25 at the time. And he happened to see this photo on the desk of Maurice Wilkins, and it is questionable whether he was supposed to have seen that, whether he took a peek when he shouldn't have, or whether Maurice showed it to him. It was not published, so it should the data, the idea should have belonged to Rosalind Franklin. Be that as it may, they had the picture for a while and didn't figure out what it really meant. And when Watson saw that photo, he knew exactly what it meant. Watson and Crick went on to determine the structure of the DNA was the double helix. They win the Nobel Prize. Wasn't determined until many years later that Rosalind Franklin played such a huge impact in that. She did not win it, and she died before anyone actually really knew that. And so she's in all the textbooks now, but I never grew up. Like when I was taking bio in high school, we did not mention Rosalind Franklin. We only knew Watson and Crick. So pretty weird deal. But I actually got to meet Watson. He came to the WashU lab when we were working on the Human Genome Project. Interesting guy. Weird guy, but interesting guy. Anyway, kind of a cool deal. We had labs named after almost all of them when I was at WashU. We had Watson and Crick and Franklin, and then we had another one, Avery, who played an important role as well in early genetics. Oh, gotta get rid of the pen. Won't let me click. So real quick on RNA, it's just a different sugar. So you have ribose instead of deoxyribose, so a little different sugar, okay, so different structure. And then single-stranded instead of double-stranded. And then there's three main types of RNA. You probably remember mRNA, tRNA, and rRNA. There's actually more than that, and we'll get to that later in the year in genetics. But RNA is actually involved in far more processes. DNA just holds the code. RNA actually does things. So a lot of scientists think RNA is far more interesting than DNA. So, oops, sorry. Again, single-stranded, not double-stranded. And remember, we are substituting uracil for thymine. So you'll notice in the strand over here, there are no T's, there are only U's. Okay, so ACGs and U's for RNA. Very easy way to tell if you're looking at RNA or DNA. Three major structural differences. There are some functional differences as well. But DNA is double-stranded, RNA is single. You have the U and the T substitution, and then the different sugar. So three big structural differences, and pretty easy ways to tell if you're looking at RNA or DNA. And here we are with the Be Sure You Can questions. So these are always good. I don't know if there's one more or not. Nope. Always good questions for you to go back and help you review and get ready for stuff like the exam that's coming up. So hopefully this was helpful. If you guys have questions, please let me know.